Bismillah ar-Rahman ar-Rahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about the DNA, which is genetic material. Okay, now this is actually a series of videos lecture will be going on in the series of videos lecture we are going to talk about, we are going to prove experimentally, we are going to experimentally prove that DNA are actually the genetic material, or they are the hereditary material. They can transfer the information from parent to offspring, from stem to cell, horizontal transfer, just like that. Okay, we'll be talking all in a series of videos lecture. So what we are going to talk about, we are going to talk about DNA. Now this is our first video lecture which are regarding the DNA as a genetic material. Okay, and in this first video lecture we are going to talk about the Griffith experiment, that Griffith can prove that DNA is a genetic material. Okay, Griffith, usually in 1928, Griffith. He proved that DNA, he experimentally proved that DNA is actually act as a genetic material, or DNA is a hereditary material. They can transfer the character from one cell to another cell, or they can transfer the character from parent to daughter. Okay, now how he proved that DNA is a genetic material. Before that, we need to understand that what are actually the genetic material. Before the genetic material, we need to understand about the genetics. Genetics are actually, it is the branch of science which deals with the study of genes, in which we talked about the genes. Okay, and we call that genetics. Now then, what are the genetic material? Genetic material are those material which can transfer the character, which can transfer the information from one cell to another cell. We call that genetic material. And usually we have DNA is a genetic material. Okay, we have also RNA is also a genetic material. But in the series of video lecture we are going to prove that DNA is actually a genetic material. Now the other thing is that hereditary material. We also say that DNA is a hereditary material. Now what does hereditary material means? Hereditary material means that DNA have the information which is present in a parent cell, and it should be transferred to the daughter cell. We call that hereditary material, and it is possible through a reproduction, or it is possible through without it is usually possible by reproduction phenomena. Now how he proved that DNA is a genetic material? In 1928, he used a bacterial strain, and he called that bacterial strain, he used a strain of the bacteria, we call that streptococcus pneumonia. So he used streptococcus pneumonia. Now streptococcus pneumonia, there are two strains of the streptococcus pneumonia. He used two strains of the streptococcus pneumonia. The one strain is smooth type of streptococcus pneumonia, and the other is rough type streptococcus pneumonia. Okay, I write it in a short form. S means smooth, T means type, S means strepto, and P means pneumonia. Now streptococcus pneumonia is it is a cocci like shaped bacteria which make a chain like structure. If I draw the structure, so it will be look like in this form. So cocci like bacteria, so these are the cocci bacteria, and they make a group or chain like structure, so we call that streptococcus. Okay, now streptococcus pneumonia are those bacteria which can cause pneumonia inside animal or inside the animal and cause the death of the animal. Pneumonia is a kind of lung disease in which the patient feel difficulties during breathing, okay, because there are some bacteria that are present in the lungs or in the especially in the alveoli area, and due to this way there is a difficulty in breathing, so we call that pneumonia, okay. So it is usually caused by streptococcus pneumonia bacteria. Now Griffith take two strain of the streptococcus pneumonia. One is this smooth type streptococcus pneumonia, another is the rough type streptococcus pneumonia. Now why we call it smooth type streptococcus pneumonia? Because in this case, in case of the smooth type streptococcus pneumonia, you know, this type of bacteria can show smooth like colony on a culture media. That's why it was called it smooth type streptococcus pneumonia. First thing. The second thing is that in this smooth type of streptococcus pneumonia, there is an extra layer or there's extra polysaccharide layer. So just like that. So this red color can show the, you know, this red color can show the layer, the polysaccharide layer, the glycoprotein layers, which we call that capsule. Capsule. And due to this way, this bacteria was virulent. Okay, this bacteria was virulent. Now, why this bacteria was virulent? Because this capsule can protect the bacteria from the phagocytic cell. So that's why this bacteria can cause virulency, or we can say this kind of bacteria can cause disease. Okay, so because it is protected their self, and that's why it was not engulfed. Now this is the first things that it was the virulent. The second thing, he also used the rough type of bacteria. You know, rough type of streptococcus pneumonia, they can show colony inside on a petri plate, just like in a rough form colony they can show, just like in this form, if we saw the structure like that. So it was called rough type rough type streptococcus pneumonia. In this case, they don't have capsule and it was non-virulent. Okay, non-virulent means they don't have the ability to cause disease. Once it is internalized, so the immunocells can detect it and the immunocell can engulf this type of bacteria, and finally the process are under the control and this bacteria cell are die. Now, so we can say that a rough type of bacteria doesn't cause the disease. Okay, this is a little bit information about the bacteria that was used by the Griffith during his experiment. Now, what actually he performed? He also take mouse. Okay, he also take mouse. Okay, this is not the exact structure of the mouse, but he take the mouse. Okay, now this is, let's suppose, the mouse. He take, you know, he take 4 mouses. Now, what actually in the first step he performed? In the first step, he used this S-type bacteria. You know, the S-type bacteria was present in a solution, and they take this solution from inside the injection, and then they, you know, inject this S-type bacteria solution inside the mouse. Okay, they inject this kind of solution inside the mouse. Then what was the result? The result was that the mouse was die. Why? As I told you that this type of cells can cause disease like pneumonia. The mouse got pneumonia and finally died. Okay, now in the next step, he take again the same mouse, and then he inject this mouse with the R-type bacteria. Okay, R-type bacteria. So the result was that the bacteria, the mouse was alive. Now why? As I told you that this bacteria don't have the ability to cause disease because they don't have the polysaccharide layer or capsule, due to which they can show the protection. As they don't have the capsule, so protection is not possible, and that's why these cells this bacterial cell was recognized by the immunocells, and finally there's no chances of the disease, and this mouse will survive and alive. Okay, now third time what actually he performed, in the third step he take the same bacteria, means the same smooth type bacteria, and this time he used, he provide heat to that solution. He provide heat to that solution which contain S-type streptococcus pneumonia. Okay, so let me write. Heat killed. We call that heat killed S-type streptococcus pneumonia. Now he take this kind of bacteria and he provides some heat to eliminate or to destroy the virulency ability or the pathogenic ability of this bacteria, or we can say they destroy the capsule area of the of this bacteria, due to which this bacteria don't have the ability to cause disease. So when they inject this type of bacteria to the mouse, so then what was the result? The result was that the mouse lived and survived. Why? Because if there is no capsule, so again it behaves like the non-virulent bacteria, because they don't have the capsule. So how they will protect themselves? So the mouse survive and the immunocell can kill this kind of bacteria. The bacteria is already killed, so then the mouse are survive. In the fourth step, what actually he performed? The fourth step was very surprising for Griffith as well as for us as well. How? This heat killed S-type streptococcus pneumonia and this non-virulent, or we can say R-type streptococcus pneumonia, both don't have the ability to cause disease in the mouse, and they don't have the ability to cause the death of the mouse. Okay, now he used both cells. He used the mixture of heat killed S-type streptococcus pneumonia and he used R-type streptococcus pneumonia. Okay, he used the mixture of R-type streptococcus pneumonia and heat killed S-type streptococcus pneumonia. And once he used the mixture, and when they inject it to the mouse, then what happened after that? After that, the mouse doesn't survive and die. How the mouse die? Because we talked that heat kill S-type streptococcus pneumonia and R-type streptococcus pneumonia don't have the ability to cause diseases. Then the mixture can cause the death of the mouse. How it is possible? This was the surprising for the Griffith. And then finally Griffith concluded that there was something transfer from this bacteria to the S-type, and he concluded that it was actually the DNA. He says that it was actually the DNA, and finally he proved that DNA is actually a genetic material. Okay, he take the the blood samples and he grow it, and finally he found this rough type of bacteria, streptococcus pneumonia bacteria. He found these kind of bacteria, and finally he proved that this kind of bacteria receive something which is just like the which is just like the S-type bacteria, and that's why this type of bacteria can cause the death of the mouse. And he concluded that actually the material was DNA. The material which was transferred to the bacterial cell, to this R-type bacterial cell, which can cause the death of the mouse, it was actually the DNA, which was transferred to that cell by the heat-killed S-type bacteria. Okay, but this experiment was somehow rejected. by Avery and Avery and his co-workers. He reject this step. Although this experiment can prove that DNA is a genetic material, but still it was rejected. Now, how this experiment was rejected? The experiment was rejected because it is not possible that inside the mixture, you may know that there is also the mixture of protein. Means this polysaccharide is made up of protein layer or glycoproteins, just like that. This there is also the RNA portion. There is also some other portion as well. So maybe there is a chances that the protein portion is also transferred. The RNA portion is also transferred. Maybe there is a chance. So then how? Then these scientists say, then how Griffith, says that DNA is a genetic material. Maybe the protein is a genetic material. Maybe the RNA is a genetic material. Because this mixture, this heat-killed smooth type of bacteria may have also the capsule, protein layer of the capsule, as well as they have also some other, substances due to which we can say that these substances are also a genetic material. But the genetic material which is transferred from this bacteria, this heat kill bacteria, to the R-type bacteria, then we call that transformation. Okay, so transformation is nothing but it is a process in which the naked DNA is transferred to the bacterial cell, and it is mixed with the chromosome of the bacterial cell, with the another chromosome of the bacterial cell. We call that transformation. Okay, there are two types of transformation: horizontal gene transfer, and we can say horizontal gene transfer, and we can say vertical gene transfer. So let me a little bit talk about that. What are horizontal gene transfer and what are the vertical gene transfer? In horizontal gene transfer, when the genetic material is transferred from the same cell, we call that horizontal gene transfer. For example, we have a bacterial cell. We call that again, let's take the example here, streptococcus pneumonia. We take the bacteria, and at that era we have also streptococcus pneumonia. Okay, so this is rough and this is smooth type of streptococcus pneumonia. So when although both are the same bacteria, okay, but the properties are a little bit different. The genetic material, when it is transferred to another bacteria, but the bacteria, the bacteria are same, we call that transfer, we call that horizontal gene transfer. Now, vertical gene transfer are those phenomena in which let's suppose in which the gene are transferred from parent cell to the daughter cell. We call that vertical gene transfer. So in this case, we saw that the genes are transferred from the one cell to the different cells. Okay, we call that vertical gene transfer. Now, so that's in a sense about that Griffith experiment, that how he proved that DNA is a genetic material. Inshallah, in the next video lecture, we are going to talk about Avery and his co-worker experiment. He also proved that DNA is also a genetic material. Okay, but he also put a question mark on the Griffith experiment, and they says that DNA is not a genetic material. Then they perform some different kinds of experiment, and finally they also concluded that DNA is actually the genetic material. Okay, now how they perform that DNA is actually a genetic material, we'll be talking inshallah in our next video lecture. Okay, so that's in essence about the Griffith experiment. I hope you get the idea about that. Thank you so much.